So good evening to the students who have joined till now. Can you let me know in the chat box if my voice is audible, and if you can see my video and also the presentation. Please confirm in the chat box that is everything okay with the voice, with the presentation. Okay, thank you so much. So we will wait for some time and let others join, and then we will start today's session. Okay, so welcome all in the today's session. We will wait for just 2 to 3 minutes more. Let other students join all too, and at sharp 6 p.m. we will start today's session. Okay. So today it is the week 2 live session for the NPTEL course of organic chemistry in biology and drug development, and the class will be up to 8 p.m. So from 6 to 8, so we will wait for 2 more minutes, let more people join, and then we will start today's session. Okay. And meanwhile, I hope my voice is clear and the video and the presentation also is visible. If there is any issue regarding the network, please let me know in the chat box. We will start shortly in just 2 minutes. Okay, I think we can start with the introductions. Okay, so I hope my voice is clear. Is there anybody who cannot listen to my voice, or I think there is everything all right with the presentation also? So if there is any issue, please let me know in the chat box. So very good evening to the students who have joined till now. My name is Mousumi Deb and I am the PMRF fellow from the Department of Chemistry and Chemical Biology, IIT Dhanbad. It is the NPTEL course of Organic Chemistry in Biology and Drug Development, for which the week 2 live session is today. So in this week 2 live session, what we will do, we will cover the week 3 course contents. So I will start the class and whenever there is somebody who wants to join, I will just let them in. Okay, so bear with me if there is any disturbance due to that. And one more thing I want to clarify, that today it is week 2 live session, but in last day we already did the week 1 and week 2 course content all together. Okay, so today what we will do, since the week 3 the course assignment and everything is live right now, so today we will solve a few questions from the week 3 class content. Okay, and these topics and these questions will help you to understand the chapters well, and it will help you to understand the topics well, and also it will help you for answering your weekly assignments. Okay, so today what we will do, we will cover few topics from the week 3, and before starting I want to explain few things about the live session for the NPTEL course. So first, that there will be a problem solving session every week. Okay, so there will be a problem solving session every week on Tuesday from 6 p.m. to 8 p.m. and the sessions will be live from the on the Google Meet platform. The link for the classroom is the same as today. So this link will be live for next already we have done 1 week, so we will have to do it for 11 more weeks. Okay, so this Google Meet link is the constant for the all classes for this course, and in between the class if any network disruption happens, I will request all the students for waiting for few minutes. Okay, do not leave the class. I will join as soon as possible. If there is any network issue regarding your side, from your side, so don't worry, the class video and also the PPT, it will be shared to you by Friday this week. Okay, so that you can just check the videos and the presentation later if you miss anything by chance during the class. Okay, and during the sessions the students can post the question and doubts through the chat box. Okay, the chat box is always open. I am checking the chat box continuously. If there is any problem regarding any question, anything you feel like telling me, you can write in the chat box. Okay, I have just muted everyone's voice and video so that there is no interruption during the streaming. Okay, since because so if you have anything to say, like you have any problem with the network or you have any problem with regarding any question, anything, you can write in the chat box. One more thing, whenever I am showing you some question, I want all of you to try to answer that question. Okay, so all the question will be like multiple choice question, MCQs, and there are 4 options for each question, and I want all of you to try to answer like which of the following is the correct answer, and write your option, what you think is the correct option in the chat box. Okay, so after each question I will check the chat box answers, and after that we will discuss the particular question and particular problem. So it is fine if you don't know what is the answer, correct answer for that particular question, but I want all of you to try. Since there is no marks answering here, you don't have to worry about it. So I want all of you to answer, because otherwise I won't understand if you are if guys are understanding the topic or not. Okay, and after the class, if still any doubt persists, the students can drop an email to me. I will answer that query by email. I will share my email ID at the end of the session. Okay, and just like I have said earlier, that the presentation and the class recording, it will be shared to the students very soon. By this Friday, I think it will be shared to you by the NPTEL website. Okay, so today it is week 2 live session, but as I have said earlier, that today we will discuss the week 3 course content of the course Organic Chemistry and Biology and Drug Development. So what are these course contents? The first thing is the hierarchical structure of proteins, secondary, tertiary and quaternary structure. Then there is a Ramachandran plot and the protein purification techniques. Protein purification techniques, it will be continued to the next chapter. The introduction to enzymes and the kinetics, the enzyme catalyzed reaction, and introduction to the catalytic activity of proteases. So these will be today's topic, based on which I have made around 20 questions, and I want all of you to answer this, try to answer the questions. Okay, and we will discuss each and every question very thoroughly. So if there is any different type of question you face in your final exam or assignment, I hope you will able to solve those questions. Okay, so I think sufficient students have joined. So again, if there is any problem regarding the voice or the video, anything, just let me know in the chat box. Okay, and if any network issue happens, please do not leave the call, just wait for few minutes, I will surely join back. So let us start. So the first question of today's session is, it says, which amino acid is incorporated into the polypeptide chain to induce flexibility, and which amino acid is termed as a helix breaker? Your options are as follows. Number A, alanine and glycine. Number B, glycine and proline. Number C, proline and glycine. Number D, histidine and proline. You have to answer it in respectively, like which amino acid is incorporated into polypeptide chain to induce flexibility, and which amino acid is termed as helix breaker. So give me a minute. Okay, so we have started getting the answers. Okay, others also try. Which of the following you think is the correct answer? Which amino acid is incorporated into polypeptide chain to induce flexibility, and which amino acid is termed as a helix breaker? Your options are number A, alanine and glycine. Number B, glycine and proline. Number C, proline and glycine. Number D, histidine and proline. Yes. Somebody wants to join. Okay, so we are we have started getting the answers. Okay, so others also try. There will be a little bit of disturbance at the beginning because many students are trying to join, so that's why I have to just change the PPT. Okay, so take time. there is no problem. I will repeat the question again. Which amino acid is incorporated into the polypeptide chain to induce flexibility, and which amino acid is termed as a helix breaker? Your options are number A, alanine and glycine, number B, glycine and proline, number C, proline and glycine, and number D, histidine and proline. So I think there are few answers in the chat box. So let me check the answer, then we will discuss this question in detail. Okay, so there is one answer, number B, and there is one answer again, glycine is helix breaker. Okay, so let us check if the answers are correct or wrong. So the correct answer for this question is number B, glycine and proline. So yeah, there was a correct answer in the chat box. So glycine is the amino acid which is incorporated into the polypeptide chain to induce flexibility, and the amino acid which is termed as helix breaker is proline. So what is a helix? At first let us discuss that. What is a helix? So helix, it is basically the secondary structure of the protein which is called the alpha helix. So as we know, what is the primary structure for the proteins is basically the polypeptide chains. Okay, so the polypeptides, the peptide linkage, the polypeptide chains, these are the primary structure of protein. So what happens when the amino acids come together? There is a carboxylic acid group in a particular amino acid and the amine group of the another amino acid. They basically react together and form a peptide bond, which is the CONH entity. Okay. There is separate thing. Okay. So this is a CONH entity which is termed as the peptide linkage. So what happens after the primary structure of the protein? There is a secondary structure of the protein. Means these polypeptide chains, they actually interact with each other via the hydrogen bonding basically. Okay, so this interaction gives rise to two type of secondary structure of the protein. The first one is termed as alpha helix, and the second one which is termed as a beta pleated sheet. So for the alpha helix, as you can see from this structure, so this is the alpha helix structure. If you can look at this position from the top, you will see alpha helix type of structure. This is called the alpha helix. Okay, and so what happens here? Here is the polypeptide chain, they stay as this kind of helix formation. There is a amine terminus is here, the carboxylic acid terminus is here. Okay, and each of the fold of the helix, the distance between the two folds is 5.4 Armstrong, and in this 5.4 Armstrong there are 3.6 residues of the amino acid are present. As you can see, there is the color for each and every element. Like for carbon, this is grey, white for hydrogen, red for oxygen, blue for nitrogen, R for the purple for the R group. Okay. So this is the kind of alpha helix. Now there are many amino acids are present in our in the polypeptide chains. So one amino acid is proline. So proline, what is the structure of proline and why it is special? Because proline is the only amino acid which has this kind of structure. Okay, the proline, it has a secondary amine group. Okay, and it is a cyclic amino acid. The structure of the proline is like this. It is a imine amino acid. Sorry for my bad handwriting here. So this is a kind of structure of proline. This is a 5-member ring, and there is a NH here and carboxylic acid group is here. Okay, and so as you can see in the alpha helix, there is this kind of hydrogen bonding. Okay, so one NH group from one turn reacts with the carboxylic group for the another turn just below of it, and there is a hydrogen bonding, and this hydrogen bonding gives stability to this alpha helix. So what will happen if there is a proline? So the proline is a secondary amino acid. So after the amide bond formation, means the peptide bond formation, there will not be a hydrogen atom like present here for the normal amino acids. Okay, since there is no hydrogen bond donor here, okay, since there is no amide bound hydrogen which can act as a amide hydrogen bond donor, what will happen? There will not, if there is a proline present in the polypeptide chain, what will happen? There will not be this kind of hydrogen bond formation with the amino acid just present just in below for this proline group. So what will happen? The helix will not get stabilized. That's why proline is called the helix breaker. and then there is a glycine. Glycine, as we know, glycine is the simplest amino acid. Okay, so what is the structure of glycine? The structure of glycine is like this. There is a CH2 group and there is a amine group one side, and there is a, this is nitrogen, okay, amine group one side, and there is a carboxylic acid group one side. Okay, so this glycine is the simplest amino acid, means there is only 2 hydrogen atoms with this carbon. Okay, there is no and this is also achiral amino acid. Okay, so what will happen, if glycine is there, it will provide the flexibility to this polypeptide chain. Okay, so proline and glycine are the exceptional amino acids. Glycine does not have any substituent, so it is much more flexible, and so whenever you want to bring flexibility in a polypeptide chain, you incorporate glycine. You will see that at flexible regions there are glycines, and if you want to bring rigidity, means you want to disrupt this kind of system which is stabilized by the intramolecular hydrogen bond, then incorporate proline, since proline cannot participate in hydrogen bonding. So proline is basically which called the helix breaker, it breaks the helix. So if some protein is going to have a going in a helical shape and suddenly changes another shape, at that changeover point proline may be present, because proline is one which does not allow any helix formation. So apart from right handed helix, there is another structure which is called the beta pleated sheet, which we will discuss later. Okay, so I think it is clear, okay, why glycine is the amino acid which introduce flexibility and why proline is termed as helix breaker. So is there any doubt for the first question of today's session? As I have said earlier, that if you have any doubt, any query regarding any question in between, just let me know in the chat box. Okay, and if you have any query, you can tell me later also, but it is better if we can just solve the query for a particular question beforehand, before moving on to the next question. Okay, so this was the problem number 1. Again, I want all of you to participate. Whenever I am showing you a problem, I want all of you to try to answer that problem. Okay, it does not matter if your answer is correct or wrong, but at least you should try to answer that particular problem. Okay, so now I think we can move toward our next question. So our next problem, the problem number 2, it is, which of the following statements is not true for the beta bands? Number A, hydrogen bond stabilizes the beta band structure. Number B, proline and glycine are frequently found in beta patterns. Number C, it occurs at protein surfaces. Number D, it gives protein linearity rather than globularity. So first read the question carefully. Whenever there is a true false kind of question, so read the question carefully. If it is asking for to identify the true option or the false option. Okay, and beta bands are another structure for the secondary structure among the proteins. Okay, just like alpha helix. So identify which of the following statements is not true, means which of the following statements is false for the beta bands. Number A, hydrogen bond stabilizes the beta band structure. Number B, proline and glycine are frequently found in beta turns. Number C, it occurs at the protein surfaces. And number D, it gives protein linearity rather than globularity. So let me check if there is any answer. Okay, we have one answer, which is number D. Others also, please try to answer the questions. Whatever you think is it may be the correct answer, just please write in the chat box. Okay, option A, B, C or D. Maybe your answer is wrong, but it does not matter, since we are not giving you marks based on these questions. It is just for your better understanding. Take 1 or 2 minutes more, then I will show you the answer, and then we will discuss what is the significance of the beta bands and what is the correct answer for this question. Okay, let me check if there are any other answers. Okay, so D, 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 B. So we have several D and we have only one B. Okay, so let us check what is the correct answer. See, that is why I want all of you to answer, because in a class if we do not have any interaction, then it will not be fruitful, right? So let me check what is the correct answer for this question. So the statement which is not true for beta bands is number D, very good. It gives protein linearity rather than globularity. Okay, so let us check what is a beta band and what is all about. So beta bands are a type of secondary structure. Okay, so these beta bands permit the change of the direction of the peptide chain to get a folded structure. It gives a protein globularity rather than linearity. Hydrogen bond stabilizes the beta band structure. Proline and glycine are frequently found in these beta turns. Beta turns often promote the formation of antiparallel beta sheets. These occur at the protein surfaces and these involve 4 successive amino acid residues. Okay, so here you can see, like, there are 4 successive amino acid residues. There is 1, 2, 3 and 4, and these successive amino acid residues, they actually form this beta band structure. Okay, so as you can see, beta bands, or it is all frequently known as the beta turn. So these are of type 2. Okay, so here you can see this is the type 1 and this is the type 2. So what is the difference between this type 1 and type 2? As you can see here, the look at this structure of this peptide bond here. As you can see the structure of this peptide bond here. Okay, the carboxylic group is downward and the amine group is upward. So this is the type 1, and if the opposite happens, means the carboxylic carbonyl carbon is upward and hydrogen is downward, then this is the type 2 beta turn. Okay, so again let us just see the what is the beta band. So these permit the change of the direction of the protein chain to get a folded structure. It gives protein globularity rather than linearity. That is why this option is incorrect. Okay. Okay. Then hydrogen bond stabilizes this beta band structure. So this option A is correct. Proline and glycine are frequently found in beta turns. Option B is also correct. Beta turns often promote the formation of antiparallel beta sheets, and these occur at the protein surfaces, means option C is also correct. So the incorrect option is number D. Okay, so very good. There were many right answers for this question, and if somebody couldn't get the right answer, it is also fine. I think now you have understood what is beta bands and what is specificity of this beta bands. Okay. So now I will move toward the next problem. So problem number 3, it says that according to Ramachandran plot, which region is sterically disallowed for all amino acids except glycine? Number A, red region, number B, yellow region, number C, white region, and number D, all of these. So this question is regarding Ramachandran plot. So try to answer this question after your I check few answers in the chat box. I will just show you the correct answer and we will discuss what is Ramachandran plot in detail. Okay. So according to this Ramachandran plot, which region is sterically disallowed for all amino acids except glycine? Number A, red region, number B, yellow region, number C, white region, and number D, all of these. So glycine, as I have mentioned earlier also, is the exceptional amino acid. Why? Because it is the only amino acid which is achiral, means there is no chirality in glycine, since the carbon of the amino acid, the backbone carbon, it contains 2 hydrogens. It does not contain any functionalities except hydrogen and amine and carboxylic acid group. So R is basically the hydrogen here. So try to answer the question. Okay, let me check if there is any answer for this question. Okay, so number C. I think we have several answers. Okay, let me check what is the correct answer and let us discuss what is the Ramachandran plot. So the correct answer is very good. It is number C, white region. So what is Ramachandran plot? Okay, so Ramachandran plot, it also provides an overview of allowed and disallowed regions of torsion angle values, and it serves as an important indicator for the quality of the protein three-dimensional structures. Torsion angles are among the most important local structural parameter that control protein folding. Third possible, so there are as you can see there is a peptide bond here. Okay, the angle between these all bonds is known as the different torsion angles. So basically there are two torsion angle for the polypeptide chain which are very important, and they describe rotation of the polypeptide backbone around the bonds nitrogen C alpha, which is called phi I, and alpha carbon and the carbonyl carbon, which is called the psi. Okay, and then there is another third possible torsion angle of the protein backbone which is called the omega. Okay, so what happens, it provides an easy way to view the distribution of the torsion angle in the protein structure. Okay, so the, as you can see, then what is the Ramachandran structure we will show you. So this is the Ramachandran plot. Okay, so a Ramachandran plot, also known as a Ramachandran diagram or a phi psi plot, originally developed by 1963 by G. N. Ramachandran. So as you can see, this is a Ramachandran plot, this square type of box. So there are various kind of regions. Okay, so there is a yellow region, there is a red region, and there is a white region. So in the yellow region, basically left-handed alpha helix sheets are more prominent. Okay, so in the red region, what happens? So yellow regions, these are outer limits. Okay, and in this phase there is right-handed, as you can see there is a four division for the different kind of angles. So and there is a scale here which denotes the angle of the phi here and the x axis, and for the psi in the y axis. Okay, and the angles vary from -180 to 180 degree. Okay, and based on these angles there are four regions here. Okay, and at this region the beta sheets are prominent. In this region the left-handed alpha helix are prominent, and here right-handed alpha helix are probable. So white region is this region basically. So this is sterically disallowed for all amino acid except glycine. Okay, red regions allowed region, namely A helical and B sheet conformation, and yellow regions is the outer limits. Okay, so again I will show you what is the phi, and phi is the angle between the nitrogen and alpha carbon. Okay, so here is the nitrogen, here is the alpha carbon, and the angle it is called the phi. Okay, and the alpha carbon and the carbonyl carbon, the angle is called the psi, and the angle between the carbonyl carbon and nitrogen NH, it is called the omega. Okay, and based on these values, what it determines when the protein gets up to the secondary structure, when it start getting folded. Okay, the polypeptide chains, based on these angles between these bonds, there are various structure of the proteins which are allowed and disallowed. Okay, and these all things we can know from the Ramachandran plot. Okay, so remember about these regions for the Ramachandran plot, it is more important. Okay, you don't need to memorize everything, but remember like this structure, remember, and remember what is all about the white region and red region and yellow area. Okay. So this was for the problem number 3. So there were all I think correct answer for this question. So very good. Now I will move toward the next problem. So problem number 4, it says that which of the following statements is false for the determination of 3D structure of proteins? Your options are: number A, best way is to crystallize the protein. Number B, NMR can also provide a static conformation of the proteins. Number C, cryo-electron microscope can directly show the image, no need for crystallization. And number D, for NMR, highly concentrated solutions are needed. So you have to identify the false statement among these 4. This is regarding the determination of the 3D structure of the protein. Your options are: number A, best way is to crystallize the protein. Number B, NMR can also provide the static conformation of proteins. Number C, cryo-electron microscope can directly show the image. There is no need for crystallization. And number D, for NMR, highly concentrated solutions are needed. So which of the following you think is the false? Write in the chat box which of the correct answer is the for this problem number 4. Okay, let me check in the chat box if there is any answer. Okay, so if you have any question, please do write in the chat box. Okay. Okay, so there are few answers for this question. So answers are B, C, D. So we have 3 different answers for this question. We have number B, we have number C, and then we have number D. So let me check if which of the following is the correct answer. Okay. So the correct answer for problem number 4 is number B. So the statement which is false regarding the determination of the 3D structure of proteins is number B. NMR can also provide the static conformation of the proteins. So before discussing this all techniques and everything, let me show you what is the importance for the 3D structural determination for the proteins. So why we will do it? Okay, what is the necessity of doing the 3D structural determination of the proteins? So the 3D structures of proteins are precious sources for the various information, like the shape and the domain structure, the protein classification, means if the protein is a globular kind of protein or is a fibrous protein, prediction of the function for the uncharacterized proteins, interaction with other macromolecules, interaction with small ligands, metal ions, nucleotides, substrates, cofactors, inhibitors, evidence for enzyme mechanism, structure based drug development, which is a very important thing, okay, for the drug discovery method, post-translational modification like disulfide bond in glycosylation, experimental evidence for transmembrane domains. So these are the various precious information which we can get from the 3D structure of the protein. Okay, so among which very important one is the structural based drug development, because if we are discussing I mean most of the drugs, they are related to any they may be related to some proteins of our body. So before designing the particular drug or any kind of medicine, we need to know what is the particular structure and what is the particular characterization for the proteins. Okay, and again the interaction with the ligand, metal ions, the cofactors, inhibitors, the uncharacterization protein, other macromolecules. So these all can be known from the 3D structure of the proteins. Okay. So there are various methods for the determination of the 3D structure of protein. So now regarding the determination of the 3D structure of proteins, there are different techniques to determine the 3D structure of the proteins. One of the best way is the X-ray crystallography. If you can isolate a crystal structure of the protein, then that is the ultimate structural evidence. Methods of determine the complete 3D structure are usually 3 types. One is the X-ray crystallography, and it is the most reliable one. Then there is NMR spectroscopy, which is a very high field NMR machine is needed. Though NMR spectroscopy you can determine 3D structure of a protein, however, there are some disadvantages of the NMR spectroscopy. You cannot handle very large molecules in NMR, but at the same time there are some advantages as well. In X-ray crystallography, if you cannot crystallize a protein, then you cannot get the crystal structure. Okay. Okay, so where were we? Yeah. So you cannot handle large molecules in NMR, but at the same time there are several advantages as well. Okay. So in X-ray crystallography, if you cannot crystallize a protein, then you cannot get the crystal structure. X-ray crystallography needs the crystal, the structure is a static one, we get the average structure of the crystal, but NMR is recorded in the solution. So what you see is exactly the dynamics of the system, but you need a highly concentrated solution to get the signal. The peaks are usually very complicated ones. You should have isotopically labeled carbon, isotopically labeled nitrogen. There are techniques to do that, but NMR you can do the structural analysis, but not for very large proteins. I mean that there is a limit up to 60 kilodalton you can do the structural elucidation. So there are various structure, there are various techniques for the determination of the 3D structure of protein. So very important one is the NMR, as I have told you, like crystallography is another very important, X-ray crystallography, and other methods which I have here mentioned, like the cryo-electron microscope is also a very good method for the structural determination of the proteins. So what is the difference between the X-ray crystallography and NMR spectroscopy is, for the X-ray crystallography you need to have a crystal for the particular protein. Since you are getting a crystal for the protein, what will happen, it is a static structure, means the crystal does not change with time, the structure which is there in a crystal, it will stay constant, okay, because it is rigid in that case. But in the NMR what happens, we have to provide a particular compound solution, it may be protein or for any compound solution we have to provide a particular solution. So in the solution the molecules can move very freely, unlike the crystal where the molecules are rigid in a particular formation, but the solution the molecules can move at their own due to their various molecular motions. So from NMR the data we are getting is not a static one, it is a dynamic data, because with time the molecules positions are getting changes and we will get a average data for that particular timeline, okay. But for crystal we have a static structure which does not change with time. So option number B, which says that the NMR can also provide the static conformation of protein, it is false, because NMR cannot do that, because NMR we have this compound in this solution and it cannot provide the static structure. And there are various drawbacks for the NMR spectroscopy because of the proteins, because the peaks it will be very complicated, since there are so many carbon atoms and there are so many nitrogen and everything, the peaks will be very much complicated. So for 1D NMR it will be very difficult to have the correct structure. So we may need to do the 2D NMR and more different kind of NMR experiments for the particular structural elucidation for the protein or kind of macromolecule, okay. So this was your problem number 4. Now let me discuss the next problem. So this question is related to NMR, since we had just discussed about it in the last slide. So it says that which of the following nuclei is the NMR inactive? Your option as 1H means proteum, number B is 13C means 13C carbon, then it is 16 oxygen, and this then 31 phosphorus. So try to identify which of the following nuclei is NMR inactive. After your guess, we will discuss about the NMR activity regarding various atoms. Which of the following nuclei is NMR inactive? So this question is basically for your from the best purely chemistry question, but you need to know it because the course is all about organic chemistry, and in organic chemistry NMR is a very important topic. Let me check if there is any answer for this question. Okay, so okay, there are few answers. I think there is number C, number B, number C. So mostly we have number C and one number B. So Pranita, if you have to say anything, you are raising your hand. I have seen it, but please can you write in the chat box, because in between the class I do not want to just unmute everybody, because there will be some disturbance. There may be some network issues also as well from my side or your side also. So please please write in the chat box whichever you think is the correct answer, or if you have to anything to know. Okay. Okay, so we have many answers for this question, and mostly the answers are telling me the correct answer will be C. So let me check which is the correct answer. So the correct answer is yes, it is number C, it is 16 oxygen. So NMR active and NMR inactive nuclei. So as you can see, it is your atomic symbol. Okay, and here on the top which we have the mass number, and hence there is a atomic number. Mass number is denoted by A and atomic number is denoted by Z. Okay, so if you have even mass and even atomic number, then we will have NMR inactive. But if both of the mass number and atomic number are odd, or one odd and another even, then we will have the NMR active molecule. Okay. So why this happens? So basically what happen if there is both of the mass number and the atomic number is even one, what happens, the I value, the magnetic moment, this can get cancel out with each other. Okay. So that's why we need to have one even and one odd, or both odd can happen. Okay. So as you can see, is there is the I. So this I is the half integer for the odd and odd atomic and mass number. Like protium, this value of I is the half. Okay. Then for the 13C, the value is odd and even, means the mass number and atomic mass is odd, but the atomic number is even. Okay. And for the so that's why we have half integer value for the I, and it is NMR active. For even and odd, the like for this example, the protium, the mass number is atomic mass is even, but the atomic number is odd, but still we have the integer or half integer value, but we will have the I equal to 0 when both of this are even. So this is NMR inactive, like 12 carbon, 16 oxygen, these has both of the atomic number and mass number even. So these have actually these are NMR inactive. Okay. So this was a basic question related to NMR, and most of you have answered it very correctly. Okay. So now I will move toward the problem number 6. So problem number 6 says that which of the following statements is false for gel filtration technique? Number A, it is also called the size exclusion chromatography. Number B, chemical polymers can also be separated by this technique. Number C, large size proteins will elute first, followed by the smaller size ones. And number D, different sized cutoff gels are used to separate the proteins. Okay, there is some mistake I think here. Okay. So I think there is something wrong with this question also, because okay, there is, okay, let me do one thing. Okay, one option was incorrect, so just give me 1 minute, because one option was incorrect here. Okay, I will stop sharing for 1 minute, because one option was incorrect here. Let me just clear it out. Just wait for 1 minute. Let me just correct this thing. Okay. So is my screen visible? Please let me know if the screen is visible. Yeah. So very sorry, there was some mistake for that question. Okay, I will repeat again the question. So which of the following statements is false for gel filtration technique? The options are: number A, it is also called the size exclusion chromatography. Number B, chemical polymers can also be separated by this technique. Number C, small size proteins will elute first, followed by the large size ones. And number D, different sized cut-off gels are used to separate the proteins. So which of the following is false regarding the gel filtration technique? Let me know in the chat box. After that we will discuss about the gel filtration chromatography in more detail. The options are: number A, it is also called the size exclusion chromatography. Number B, chemical polymers can also be separated by this technique. Number C, small size proteins will elute first, followed by the large size ones. And number D, different sized cut-off gels are used to separate the proteins. So is there any answer for this question? Okay, number C. Okay, again Madhura, if you have anything to tell me regarding the PPT or anything, please let me know. Okay, the problem is not visible to me. Okay, is the problem is visible to everybody else? I had actually off the slide sharing, but then I had just shared it again. Others, please let me know if the PPT is visible to you or not. Okay, so I think Stephen, I think there is something wrong with your network issue. You can just leave and rejoin. I think then the problem will be visible to you. Since I had just off the PPT and then on, I think there is some issue, but you can rejoin if you cannot see the PPT, and others I think everybody is told that it is visible to them. So I think you can rejoin. After rejoining you will see the PPT, and if you cannot see the PPT, don't worry, I will share it later. So but I think rejoining will solve your problem. Okay, so let me show you the answer. So the correct answer means the false statement regarding the gel filtration technique is number C, small size proteins will elute first, followed by the large ones. So I did the mistake because I just forgot to correct it. I mean, I had given you the right answer, but I forgot to make change in the questions. Anyways, don't worry. So let's discuss what is the gel filtration technique. Okay, so gel filtration chromatography. So what is a chromatography? So let us discuss what is the technique of chromatography and why it is so much important in a particular synthetic lab or anything. Okay, so chromatography encompasses a variety of techniques that involve a mobile phase flowing through a bed of the stationary phase. A mixture of solutes is resolved and or separated as a result of such differences in affinity of the solution for the mobile phase relative to the stationary phase. A solute that has more affinity for the mobile phase move forward with the mobile phase, whereas a solute which has more affinity for the stationary phase is retarded. Thus, solutes are separated into the bands based on their distribution between the phases. Molecules of the biological interest are separated or resolved by variety of chromatography techniques, including adsorption chromatography, ion exchange chromatography, partition chromatography, which may be paper or thin layer chromatography, which is known as TLC, Grash liquid chromatography, and molecular exclusion chromatography by molecular sieve or gel filtration. So chromatography, it mainly this is a kind of chromatography. So what happens in chromatography? Let me tell you in brief. So what happens in chromatography? So basically in chromatography what happens, there is a stationary phase, and most often and most commonly the stationary phase is the silica gel. Okay. So what happens in thin layer chromatography? So most commonly used chromatographies in organic chemistry or synthetic chemistry is thin layer chromatography, which is known as TLC, and then is a column chromatography. So what happens in the thin layer chromatography, there is a on a particular there is a paper kind of thing, then after on the top of the sheet there is a thin layer of a silica gel, and what happens after the this papers are cut in a very small portions, and we have to put the concentrated solution of a mixture on as a dot on that particular paper. So I will show you actually what happens. So in thin layer chromatography, a layer of a silica gel is used. Okay, so there is a particularly, I think there is a this kind of, okay, I just tilted it. So there is a this kind of thing. Let me just erase it a little bit. Yeah, so in thin layer chromatography there is the thing called TLC plate. Okay, and what is this TLC plate? There is a particular silica a sheet on the top of which there is a thin layer of silica, and we have to give there at the bottom there is the 3 or 4 dots. How many compounds are there? You have to put the solution in a dots. Okay, and this silica gel sheet is called the stationary phase, and then we after you put the solution as a of a compound as a particular a little drop with the help of capillary, you will add the solution here. Okay, and let it run at this position, and the flow of the solution is upward. So what happens, the compounds they actually based on their affinity to this silica gel, they show spot on this TLC plate. So if there is a compound, they will show different spot on this TLC plate, and the distance travelled from this to this is called the solvent front, up to the which the solvent is there, and the distance between these is called the RF value. Okay, and this is particular based on this RF value. What happens, we come to know that how much the compound has the affinity for the silica gel. So if the compound has a very lower RF value, means it is very polar, means it will stuck with the silica gel soon, and if the compound has a very large RF value, then with the compound what will happen, it will have less affinity toward the silica, means the compound it is the more non-polar. Okay. So what happens, like in the chromatography, then this is called the, and size exclusion chromatography is one type of chromatography. Okay, so the this TLC is for the only for the visualization. So another technique is called the column chromatography. So as you can see in this picture, there is a particular this cylindrical shape of the apparatus which is known as the column, and in this column there is a your stationary phase. Okay, so basically in gel filtration chromatography or size inclusion chromatography, molecular sieves are used for the stationary phase. Okay, and then as you can see there is a stationary phase, and when you are just giving the solute and the mobile phase, what happens, if the particular solutes they have more affinity toward the stationary phase, they will keep stuck in the particular column, but if they had more affinity toward the solution phase, they will just go with the flow, and they will just came out of the particular chromatography. Okay, so as you can see here, let us focus on this part. So here this is stationary phase, and this stationary phase are molecular sieve for the particular size exclusion chromatography. And as you can see, there is a blue color large molecules which are unable to enter the pore for this particular stationary phase, and but the small molecules they can enter the particular stationary phase very easily. Okay, since so what will happen if you are keeping this combination for the large and small molecules in your sample, and you are just adding the mobile phase, what will happen, the small molecules they will able to enter the pores for the stationary phase, and they will remain stuck in the stationary phase. Okay, and they will not come out very easily from the particular column, and the larger molecules they will not they are unable to enter the pores for this particular stationary phase. So what will happen, the large molecule they will come out fast from this apparatus, and the small molecules they will come out slow. So if you put a graph for this particular retention time, so what you will see, the retention time for the large molecules it is lower than the retention time of the small molecule. Retention time is how long the compound is there in the column. Okay, so that is the particular retention time. So that is how you will understand. So the option number 3, what it said that small molecules, small size proteins will elute first, followed by the large size ones. So this is the false question, because first the large molecules will come out of the particular elute first and then the small molecules. Why? Because the small size protein have a greater affinity toward the stationary phase. Okay. So here you can see chromatography involves a mobile phase. It can be liquid or gas flowing over a stationary phase, a solid or liquid. A mixture of solutes is separated based on the solute components degree of affinity toward the mobile and stationary phase. If compound number 1 has a stronger affinity for the stationary phase than compound number 2, then compound number 2 will migrate through the column more rapidly than compound number 1. Okay? So this is all about the gel filtration chromatography. So now I will move toward the next problem. It was on a problem number 6. So now problem number 7, it says that which of the following statements is true for the SDS polyacrylamide chromatography. Your options are number A, here SDS stands for sodium dodecyl sulfate. Number B, SDS is used to unfold the proteins to make them linear. Number C, SDS binds to the proteins non-covalently with the stoichiometry around 1 SDS molecule per 1 amino acid. And option D, all of the above statements are true regarding the SDS polyacrylamide chromatography. Try to answer this question. Which of the following statement is correct regarding the SDS polyacrylamide chromatography? It is also known as SDS page. SDS page chromatography is also known as. Let me know in the chat box which of the following you think is the correct answer for the SDS polyacrylamide chromatography. Option A, here SDS stands for sodium dodecyl sulfate. Number B, SDS is used to unfold proteins to make them linear. Number C, SDS binds to the proteins non-covalently with a stoichiometry of around 1 SDS 1 molecule per 1 amino acid. And number D, all of the above. Number D. Okay, so there are most of the options are number D. Okay, so let me check which is the correct answer. So the correct answer is yes, very good, it is number D. So purification of proteins by SDS page. So this technique involves loading a sample of your mixture onto a polyacrylamide gel which is called the page. Okay, polyacrylamide works as like agarose except the matrix has a smaller pores and so polyacrylamide gel separates smaller molecules like proteins. Agarose is used for much larger molecule for like DNA and RNA. Unlike DNA, RNA proteins do not have a nice constant charge to mass ratio and can have any charge at a given pH depending on the sequence of the pi. To overcome this problem, proteins are coated with a detergent named SDS to make it negatively charged. Then they separate by the molecular weight. The SDS will disrupt the secondary tertiary structure of the subunits will separate. For this region, SDS page separates the subunit molecular weight. So what happens, like based on the pi or the isoelectric point which we discussed in the very last class, okay, that based on the pH and the different amino acid will have different charge. At some particular pH they will have negatively charged, or at a particular pH they will be neutrally charged at the pi, and at the particular pH they will have positive charge. So since they have different charge depending on the sequence of the separation gets complicated. Okay, so that's why we have to use this SDS which is a detergent, and the full form is like sodium dodecyl sulfate. So what happens, like as you can see, it is the folded structure, okay, of the protein. and the protein is folded with the negative and positive charges. There is positive charges, there are negative charges. Okay, so what happens? And SS is stand for the disulfide bonds. So first it is reduced. Okay, the disulfide bonds are reduced by 2 mercaptothiol. Disulfide bond are reduced from SS to SH. Okay, and then SDS, it comes with a negative charge, and this SDS makes this protein completely negatively charged and unfolded to linear structure with negative charge proportional to the polypeptide chain length. Okay, and then what will happen? Then we have to run this particular purification process, and based on the molecular weight, this protein they will get separated. Okay, and there is molecular weight markers as you can see from this picture. So there will be molecular weight structures. So again I will show you the options which were there present in the question. So option number A, SDS stand for the sodium dodecyl sulfate, it is true. Number B, SDS is used to unfold the proteins to make them linear, it is also true. SDS bind to proteins non-covalently with stoichiometry around 1 SDS molecule per 1 amino acid, this is also true. So the correct answer will be number D, since all of the above answers is true. Okay. So this was about your SDS polyacrylamide chromatography. Okay, so I will show you the next problem. So it is almost 7 p.m. So I will give you a 2 minute of break. I will show you the problem number 8, then I will give you 2 minutes to solve the problem number 8. I will not move, I will not leave the class, I will be there, but I will keep my camera and microphone off for 2 minutes, and after 2 minutes I will just again I will tell you the correct answer for the problem number 8. So first let me show you the particular problem. So problem number 8, it says that which of the following statements is true about 2-dimensional electrophoresis. Your options are: number A, proteins are separated on the basis of the charge. Number B, proteins are separated on the basis of their molecular weight. Number C, proteins are separated on the basis of the pi. And number D, proteins are separated on the basis of both molecular weight and pi. So this problem number 8, I want all of you to read the question carefully and try to answer it in the chat box. Okay, I am not leaving the class, I am just keeping my camera and microphone off for just 2 minutes, and when I will just check after 2 minutes, and I want all of you to try to answer this question. Okay, it does not matter if your answer is correct or wrong, but please try, and please do not leave the class in between. Okay, please do not leave the class, because I will just check the answers and we will resume the class for another 1 hour. Okay. Hello. So I have rejoined. So is my voice is audible to you? And also if my video is visible? Somebody can you confirm? Yeah, thank you Shreya, thank you Pranita. So we will just resume our class, okay. So I think there are few answers for the particular question which I have just shown you, and most of the answers are telling the correct answer will be number D, right. So let us check what is the correct answer, then we will discuss about it. So for problem number 8, the correct answer is very good. It is number D. Proteins are separated on the basis of both molecular weight and pi. Pi is the isoelectric point, okay, which we discussed in the last class. If you have missed the last class, you don't need to worry. They are on the NPTEL website at the for this NPTEL course Organic Chemistry, Biology and Drug Development. When there is a assignment page and the announcement, there will be the particular this video, and for the last class, and the PPT is shared with you. You can go back and just check about this, okay. Okay, so what is the principle for the 2D chromatography, 2D electrophoresis or 2D gel electrophoresis? So in 2D gel electrophoresis, proteins are separated as per isoelectric point and protein mass. Separation of the proteins by isoelectric point is known as isoelectric focusing or IEF. When the gradient of pH is applied to a gel and an electric potential is applied across the gel, making one end more positive than other. At all pH values other than the isoelectric point, proteins will be charged. If they are positively charged, they will be pulled toward the negative end of the gel, and if they are negatively charged, they will pull toward the positive end of the gel. The proteins are applied in the first dimension will move along the gel and will accumulate on the isoelectric point, that is the point at which the overall charge on the protein is 0, means the neutral charge. In separating protein by mass, the gel treated with sodium dodecyl sulfate SDS along with other reagent like SDS page in the 1D, this denatures the protein, that is that unfold into the long and straight molecules, then and binds the number of SDS molecules roughly proportional to the protein's length. Because at protein's length when unfolded is roughly proportional to its mass, since the SDS molecules are negatively charged, the result of this, that will be proteins will have approximately same mass to charge ratio to each other, okay. So look at this picture. I think it will be easier for you to understand by looking at the picture. So what happens, first there is a particular gel present here, okay, and then separation of this around this gel, it happens by isoelectric focusing. So first dimension, it happens the isoelectric focusing. This is a decreased pH pi range, okay, and based on the charge of the amino acids, they will actually get separated from in this particular gel, okay. And after the isoelectric focusing, the gel is placed on a SDS page gel, okay, here it will be placed, and then isoelectric focusing gel is placed on the and then the gel electrophoresis will happen, and then decreasing molecular weight, it will again be separated. So first the separation happens on the perpendicular side, then this actually what happens on this vertical side, then it makes it, we will make it horizontal, then we will just again separate it. Since it is happening in 2 dimension, it is called the 2D gel electrophoresis, okay. So most of the all of you, I think have given the correct answer for this, which is very good. So now I will move toward the problem number 9. Problem number 9, it says that in ion exchange chromatography, if the column is negatively charged, then which of the following statement is true? Your options are number A, proteins with pi less than pH of the buffer will elute. Option B, proteins with pi's greater than pH of the buffer will elute. Option C, proteins with pi equal to pH of the buffer will elute. And option D, none of the above. So what happens in ion exchange chromatography, if the column is negatively charged, then which of the following statement will be true? Proteins with pi less than pH of buffer will elute. The proteins with pi greater than pH of buffer will elute. Proteins with pi equal to pH of the buffer will elute, or none of the above is the correct option. Let me know in the chat box which of the following you think is correct. Then we will discuss about the ion exchange chromatography and how the separation happens in this particular technique. So, let me check if there is any answer for problem number 9. Okay, so your options are number B. So, I think number B is the most given answer for this. Okay, let me check and then let us discuss about the ion exchange chromatography. So, the correct answer is yes, the correct answer is number A. So, proteins with Pi less than pH will of the buffer, it will elute. Now, let us discuss what is ion exchange chromatography and why number A is correct. Okay, so this is the, let us focus on this part at first. So, what happens in ion exchange chromatography? So, here there will be the particular column or particular chromatography stationary phase. So, there the stationary phase will have a particular charge in the ion exchange chromatography. Okay. So, what will happen? The sample it will contain the positive and negative charge. Okay. And if let us say like the column or the particular stationary phase is negatively charged. So, if the stationary phase is negatively charged, so what will happen? If the stationary phase is negatively charged, what will happen if the negative and positive charge you add? So, positively charged protein will with immobilized anion surface, it will so positively charged ions proteins will be immobilized on the anionic surface. Okay. And then what we will have to do? Somebody wants to join. Okay. Yeah. So, just think about it. If your stationary phase is negatively charged and if you are giving a mixture of negative and positively charged, so what will happen? In the mixture, when there is a positive charge, it will feel more attracted toward the negatively charged stationary phase. Okay. Then what will happen? The positive charge, the compounds with the positive charge, it will get stuck in the stationary phase, and the negatively charged it will get repelled, and it will elute first the negatively charged, and the positively charged it will elute at the later. Okay. So, what happens now? Think about it, when Pi is less than pH. Okay. So, if Pi is less than pH buffer, then the protein with it will be negatively charged. Okay. And it will not bind to the column but elute. But when the Pi is greater than pH, the buffer will be positively charged and it will not bind the column. So, let me show you here. So, we all know isoelectric point which I discussed in last week. Pi is the pH at which there is a neutral charge, means it is 0. Okay. Now, if the pH it is less than the Pi. Okay. So, suppose let me tell, like for the in last week we did some examples, right, about the Pi and pH. So, let us say the Pi of a particular amino acid is 5. Okay, the Pi of the particular amino acid is 5, and if what happen, if the pH is greater than the Pi, okay, let us the pH is 6. So, what will happen? The particular amino acid it will be negatively charged. Okay. So, when the Pi is actually less than the pH, remember this, so when the Pi is less than the pH, the buffer the pH of the buffer, the protein will be negatively charged, and if the column is the particular column is also negatively charged, what will happen? Negative will repel each other and they will elute. But proteins with Pi greater than pH, what will happen? The proteins will be positively charged and they will bind to the column and they will not elute first. Okay. So, that is the difference. I mean, so the problem number, the option number A, which says that the pH is Pi is less than pH, the buffer will elute, it is the correct answer. Okay. So, these things might get you might get confused regarding this pH and PI. That is why in last week I actually tried to give you more examples regarding the pH and PI, and we also discussed about the few questions like what is the PI and particular pH, what will be the charge, we discussed in the last week. Okay. So, you can just go back to the last week's PPT and you can check from there. Okay, it is mentioned there clearly with various examples. Okay, so don't get confused between it. So, remember, when PI or isoelectric point is less than the pH of the buffer, that protein will be negatively charged, and if the column is also negatively charged, negative negative will repel and the particular protein will not get stuck with the column and it will elute first. Okay. So, this was the problem number 9. For this I will just suggest you, please go back to the last week's PPT. Okay, it is shared to you, I think, and if it not shared to you, you can write back to me, I will just share to you, and if you can see the last week's PPT, you will see that it is mentioned there clearly about the pH and PI, how the amino acids will be charged. Okay. Now, problem number 10, it says that which of the following is not a weak interaction? The options are number A, pi-pi stacking, number B, hydrogen bonding interaction, number C, salt bridge interaction, number D, disulfide bond. So, which of the following is not a weak interaction? Write in the chat box which of the following you think is not a weak interaction. Is it pi-pi stacking or hydrogen bond interaction or salt bridge interaction or disulfide bond? Which is not a weak interaction? Let me check if there is any answer. Okay, number D, number B. So, we have one number D and we have one number B. Okay, let me check which is the correct answer, which will not be the, okay, another number D. Let me check. So, the correct answer is, very good, it is number D, disulfide bond. So, disulfide bond, so let me show you first what is the every interaction looks like. So, for pi-pi stacking it looks like this. So, as you all know, this is the benzene ring. Okay. So, what is the pi-pi stacking? It is basically between the interaction between the pi electrons of the particular system. So, as you can see, we all know there is p orbitals here, right. In the particular benzene ring there is a p orbital here, the pi electron cloud, which is given rise by this p orbitals, and the interaction with this particularly p orbitals at each and every carbon is all called the pi-pi interaction, the interaction between the pi electron cloud. So, as you can see, there is no such a particular bond here. Okay. It is basically just a interaction between two different charge clouds. Okay. There is no actual bond formation, there is neither actual electron donation or electron acceptance. So, there is a not particular bond or electron share. Okay. Here this basically the interaction between two electron clouds. So, pi-pi stacking is not a strong interaction, it is it is a very weak interaction. Okay. Now, we have several other interaction is mentioned here. One is, okay, somebody wants again to join. So, we have various interactions mentioned here. Second one is the hydrogen bonding interaction. So, what happens in salt bridge and hydrogen bonding interaction? See, the hydrogen bonding interaction, what happens? There is it is between electron deficient hydrogen atom and a electron rich heteroatom. Okay. So, in here the carboxylic acid, the oxygen is O minus charge and this hydrogen is this oxygen is delta delta negative and this hydrogen is delta positive, and this interaction between this delta positive hydrogen and this oxygen atom, it is called the hydrogen bonding interaction. And what is a salt bridge interaction? It is basically the interaction between this positive and negative charge. This amine NH3 group's positive charge and this carboxylate group's negative charge, this electrostatic interaction is called the salt bridge interaction. So, all these three interactions, they are not actual bonds. Okay. These interactions are nothing, but a normal interactions. Okay. The interaction between the charges and everything, these are not the actual bond. Okay. But what about the disulfide bond? If you see, disulfide bond is basically the bond between two sulfur atom, okay, like this SS bond, and this disulfide bonds are seen between the two cysteine residue. So, as we know, cysteine has a side chain of CH2SH, a thiol moiety is there, and when the both thiol moieties react with each other, they form a disulfide bond. This disulfide bond is not a weak interaction. Why? Because it is a proper covalent bond. There is a electron sharing between two sulfur atoms. Okay. The actual bond is present here in the disulfide bond. Okay. That is why it is a very strong interaction. It is not a weak interaction. Okay. So, this was the problem number 10. Now, quickly I will move toward the next problem. Now, problem number 11, it says which of the following statement regarding enzyme is not true? Option A, enzymes are one kind of proteins. Option B, they can change the rate of the reaction. Option C, they get consumed in the course of the reaction. And option D, they are site specific in nature. So, enzyme, we all know about the enzymes, what is the role of enzyme and catalyst. Enzymes are basically nothing but a catalyst of the biological systems. So, just read the options carefully and tell me which of the following statement you think is not correct regarding the enzyme. Option A, enzymes are one kind of protein. Option B, they can change the rate of reaction. Option C, they get consumed in the course of the reaction. And option D, they are site specific in nature. So, is there any answer for this particular question? So, option C seems to be the most frequent answer. So, let me check. So, yeah, very good. Option C is not correct, which says that they get consumed in the course of the reaction. So, how enzymes work? So, here I have a little cartoon here. So, if it is enzyme and if it is the substrate. So, what happens? The enzyme, it first forms, this is the active site of an enzyme, where the substrate binds and the enzyme substrate complex is formed, and after the reaction happens, the products are formed and they get detached from the enzyme. But in this course, the enzyme is not consumed. Okay. So, the first statement, enzymes are one kind of protein, it is correct. They can change the rate of reaction, it is also correct. They get consumed in the course of the reaction, it is not true, but they are site specific in nature, it is also correct. So, what is enzyme? It is a protein with catalytic properties due to its power of the specific activation. Biological systems are sensitive to temperature change. Increase the rate of reaction without increasing the temperature, it happens with the help of the enzyme. So, particularly you can change the rate of the reaction by increasing the temperature, but biological system, basically the proteins and all these biomolecules are very much susceptible and very much sensitive toward the temperature. So, enzymes are those biological catalysts which help to increase the rate of the biological reactions without increasing the temperature. Increase the rate by lowering the activation energy of the particular reaction. They create new reaction pathway. They are globular proteins with 3D complex structure, and the active site where the reaction catalyzed is the enzyme active site is where the reaction is catalyzed. Okay. So, this was regarding the enzymes. Now, problem number 12, it says which of the statement is false regarding the induced fit hypothesis. Number A, substrate specificity range is very high. Number B, the active site is rigid. Number C, more accepted molecule model for the enzyme substrate complex than lock and key. And option D, conformational change occurs in the enzyme and the substrate. Which of the following statements is false regarding the induced fit hypothesis? Number A, substrate specificity range is high. Number B, active site is rigid. Number C, it is more accepted model for enzyme substrate complex than lock and key. And option D, conformational change occurs in the enzyme and substrate. Tell me in the chat box which of the following statement you think is false regarding induced fit hypothesis. Okay, let me check. Okay, I think I have multiple answers for it. Number B, number D, number C, number B. So, number B, C and number D, it is the most given answers. Okay, let me check which is the correct answer. So, the false statement regarding induced fit hypothesis will be number B, the active site is rigid. It is the false statement. Now, why and what is the induced fit hypothesis? So, basically it is another particular enzyme substrate complex model other than lock and key. Lock and key was the first complex the theory regarding the enzyme substrate complex. So, in the lock and key theory, it was said that a particular enzyme works with a particular substrate, just like a lock and key. Let us think the enzyme as a lock and the substrate is a key. So, in that particular substrate will bind with a particular enzyme and vice versa. Okay. But what is said in the induced fit model, like it is more, the active site is more flexible. Okay. In the lock and key theory, the active site of the enzyme is particularly rigid. Okay. It does not change with anything. But during the induced fit model, the active site is more flexible. So, what will happen if here you can see the substrate, the shape of the substrate is like this and the shape of the active site is like this. Okay, and it is very clear that it will not fit in the enzyme. So, at the vicinity of the substrate, the enzyme active site will change its the shape of the active site by the few conformational changes. Okay. So, what will happen then, after the particular change, the substrate will get fit in the particular active site and the product will form. So, the active site here is more flexible. Okay. In the vicinity of the substrate will induce the conformational changes in the active site of the enzyme and then it will get fit and the product will form. And it is the more accepted model for the lock and key theory. Okay, because it happens actually the proteins, they are enzymes are nothing but a protein, right. So, in the presence of a substrate, the protein, it will change its conformation a little bit so that the substrate can particularly bind with the enzyme and the product will form. So, here is the difference from the lock and key theory, which says the active site of the enzyme will be more rigid, but here it will be more flexible. So, number B, it is the incorrect option for false statement regarding the induced fit hypothesis. Okay. Now, problem number 13, it says which of the following statement is not true for the Km and the Vmax. Vmax is the maximum rate at which particular enzyme catalyzed reaction can proceed. A small value of Km tells us that the enzyme binds strongly with the substrate. Number C, Km is the concentration of the substrate at which the rate of the reaction reaches Vmax. Number D, at large value of Km tells us that the enzyme shows little specificity for the given substrate. So, what is the correct statement regarding the Km and Vmax? I will show you in detail what is the Km, what is the Vmax, but before that I want all of you to try this particular question. Which is the following statement is not true regarding Km and Vmax? Options are number A, Vmax is maximum rate at which particular enzyme catalyzed reaction can proceed. Number B, a small value of Km tells us that the enzyme binds strongly to its substrate. Number C, Km is the concentration of substrate at which the rate of the reaction reaches Vmax. And number D, a large value of Km tells us that an enzyme shows little specificity for a given substrate. Let me check which is the correct answer. Okay, so there are answers like number B, number C. So we have 1 number B and 3 number C. Okay, let me check which is the correct answer. So yeah, the correct answer is number C. Km is the concentration of the substrate at which the rate of the reaction reaches Vmax. This statement is not true. Why this statement is not true? And what is the Km and what is the Vmax? Let us discuss. So basically, for an enzyme catalysis reaction, there is a very important and a very famous equation, and which is called the Michaelis-Menten enzyme kinetics. Okay, so this is the particular formula for the enzyme kinetics. Okay, so V0 is the initial velocity, which is the unit is mole per time. Okay, and the S is the substrate concentration in molar. Vmax, it is the maximum velocity of the particular enzyme catalyzed reaction, and Km is the substrate concentration at half Vmax. So here number C, it says that Km is the concentration of the substrate at which the rate of the reaction reaches Vmax. It is not true. Rather, Km is the concentration of the substrate at which the rate of reaction reaches Vmax by 2 or half Vmax. So as you can see here, in the x axis we have the substrate concentration. On the y axis we have the it is the reaction coordinate, where we check the velocity of the reaction. Okay, so Vmax is the maximum rate at which the particular enzyme catalyzed reaction can proceed. So let us say here it is the Vmax. Okay, here V0 is equal to Vmax, which is the particular maximum rate of the particular enzyme catalyzed reaction. Okay, and the substrate concentration, which is at the half of the Vmax. So if it is the Vmax, it is the Vmax by 2, and this red is the red curve, it is the it is what your curve for the enzyme catalyzed reaction. Okay, so at the half Vmax, the if you extrapolate this line from the half Vmax, and you will reach on this curve, and then if you draw a perpendicular on the x axis, you will get a particular substrate concentration which is related to this half Vmax, and it is called the Km or Michaelis-Menten constant. Okay, so this is the enzyme catalyzed reaction, and it is at the very high concentration of the substrate. The equation becomes like this: V0 becomes Vmax. Why? Because at a very large concentration of the substrate, what happens? At a very large concentration of the substrate, from this equation you can see, if the substrate concentration is very high, means the concentration of S is much greater than the Km. So what will happen in this equation? You can just omit this Km. So and after omitting the Km, I mean ignoring the Km, rather in particularly comparing to S, but then S get cancelled and V0 becomes Vmax. And when the delta S or substrate at the lower substrate concentration, okay, the when substrate concentration is very less than the Km, what will happen? You can ignore this concentration of S in the denominator, and you will get this kind of equation. Okay, and you can see this equation at the initial stages. When the substrate concentration is low, Vmax depends on the substrate concentration, but at the high substrate concentration, what happens? Vmax V0 becomes Vmax, which is independent of substrate concentration. Okay, so at high substrate concentration, the rate of the reaction does not depend on the particular concentration of the substrate. Okay, so this was about the Km and Vmax. Now I will move toward the next problem. So this was the problem number 13. So problem number 14, it says that in Michaelis-Menten equation, at high substrate concentration, the rate of the reaction becomes number A, second order with respect to the substrate concentration, Number B, 0 order with respect to the substrate concentration. Number C, first order with respect to the substrate concentration, and number D, 0 order with respect to the enzyme concentration. I just discussed this thing. If you think think carefully about the curve which I just show you in the previous slide. If you understand that part, you can answer this question correctly. Read the question carefully. It says at high substrate concentration, rate of the reaction becomes the second order with respect to substrate concentration, 0 order with respect to the substrate concentration, first order with respect to the substrate concentration, and 0 order with respect to the enzyme concentration. Which of the following is correct? Think about the graph which I just showed you in the previous question and then answer this question. Let me check the chat box if somebody has answered it or not. Number C. Okay, so I have 4 number C. Let me check. Okay, the correct answer is number B. I had said that look at the question carefully. It says high substrate concentration. See, this is the graph which I have shown you earlier. Okay. So this is the substrate concentration on the x axis and the velocity of reaction goes to the y axis. Now the question reads that in the Michaelis-Menten equation at high substrate concentration, rate of the reaction becomes. So at the high substrate concentration means what? Means at this area. So in this region V0 becomes Vmax, and when V0 will become Vmax, there is only one parameter Vmax, there is no concentration of the substrate is there. Okay. So that is why the rate of the reaction will become 0 order with respect to the substrate concentration, not the first order. Okay, the first order with the substrate concentration, the first order kinetics, it will be followed at the lower substrate concentration. Okay. Let me show you once more the previous slide. Okay, this one, no, here. See, here I mentioned, right, like if there is, this is your equation. Now that question it had asked like at the higher substrate concentration. So when the substrate concentration will be very high, okay, so what will happen? We can ignore the Km in the denominator. If we ignore the Km in the denominator, so what will be the equation? I will just show you in the next slide. So our equation was V0 equal to Vmax, I am writing it as Vm in substrate concentration divided by Km plus your substrate concentration, right. And if the substrate concentration is very high than the Km, then what we will do? We can ignore this Km, okay, and if we ignore the Km, what will happen? This S and this S will be cancel out, and V0 will be equal to your Vmax. When V0 will be equal to Vmax, this reaction will not depend on the substrate concentration anymore. So at high substrate concentration, the reaction will become 0 order with respect to the substrate concentration. So understood? Remember, always remember this particular graph and then correlate with the equation. You don't need to actually you need to memorize this particular equation because this equation is very important, and if you can remember this equation, just try to see what you can do with the equation and read the question also carefully. So remember, at high concentration of the substrate, almost all enzyme get occupied by the substrate. So when there is substrate is very high, in high concentration, all the enzyme will be occupied with the substrate. means all the enzyme will be saturated. Now the reaction rate will not depend anymore upon the substrate concentration, but when there is a lower concentration of the substrate, what will happen, the number of substrate molecule will they will actually depend they will actually control the number of the enzyme substrate complex. Higher the number of enzyme substrate complex, higher will be the rate of the reaction, which follows the first order kinetics, which is in this position. So here it is the first order kinetics, here it is the first 0 order kinetics, and it in this place of the graph it is the mixed order kinetics. Okay. Now problem number 15, it says that identify the plot of ES versus time in the following enzyme catalyzed reaction of following Michaelis-Menten kinetics. ES is the enzyme substrate complex. Okay. So here it is the substrate, and we have the enzyme, and then we will have the enzyme substrate complex, and this enzyme substrate complex will change with time and it will form enzyme and product. So what is the particular graph will be for this particular reaction with time, how the concentration of the enzyme substrate complex changes with time for this particular reaction. The graphs are number A, this, number B, number C, and number D. Identify the correct answer. Let me check in the chat box. Okay, number C. I have one number C. Okay. Okay, let me show you the answer, then I will discuss. So correct answer will be number B. Okay, so look at here carefully. Why number B? So when at first there is only substrate and your enzyme substrate complex is forming. Okay, so how the concentration of the enzyme substrate complex will depend on the with time. So initially it will increase. Okay, when there is the initial only substrate and enzyme. So first what will happen, enzyme substrate complex will be forming. Okay, and this reaction is reversible. Okay, so first there will be a steady increase of the concentration of this enzyme substrate complex. After some time, when there is a sufficient number of enzyme substrate complexes are formed, the product will start forming. Okay, and the enzyme will revert back, and after this time at a certain point there is called a steady phase. Okay, and at this steady state what happened, like this the rate of the formation of the enzyme substrate complex becomes equal with the rate of the formation of the product. So the number of after certain time 10 enzyme substrate complex are forming, okay, per minute. So and 10 enzyme substrate complex are breaking to form the product. So what will happen, so this will become steady, and after some time when there is a substrate molecule, it is exhausted, what will happen, the concentration of enzyme substrate complex will fall. Why? Because the only products is forming right now, no substrate molecule is remaining, so no formation of the enzyme substrate complex. So then it will gradually decrease. So we will have this kind of a plateau. Okay. So the option B was the correct answer, this one. So now I will move toward the next problem. Problem number 16, it is a mathematical problem and it is a very easy mathematical problem. What is the ratio of V0 by Vmax when S equal to 7 km? I had shown you the equation for this particular enzyme Michaelis-Menten kinetics, okay, in the last to last slide. Use that particular equation and calculate it. Okay. Take 2 minutes and use that particular equation and calculate this particular question. When concentration of the substrate equal to 7 km, what is the ratio of V0 by Vmax? Options are number A 7 by 8, number B 1 by 7, number C 5 by 7 and number D 8 by 7. It is a very easy, just to put write the reaction of the enzyme Michaelis-Menten kinetics, put the value of substrate concentration in that particular equation and then just make few changes, then you will get the answer. Let me check if somebody has done it. Number C. So is this number C for which one? I don't know. I think it was for previous one. Okay, let me show you how it is solved, although it was a very easy question. Okay, so the correct answer will be number A, 7/8. So now how this kind of problems are solved? So basically this is the Michaelis-Menten equation, V0 equal to Vmax into substrate concentration divided by Km plus substrate concentration. Okay, so here what is it is given, like substrate concentration is 7 km. So what will happen if I put the substrate concentration as 7 km? So if I take Vmax here, okay, and it will become V0 by Vmax equal to substrate concentration divided by Km plus substrate concentration. Now replace the substrate concentration as 7 km. If you replace the substrate concentration with 7 km, so what we will have? We will have on the numerator 7 km and in the denominator we will have km plus 7 km. So total of 8 km. So the ratio will be 7/8. So that is how this kind of problems are solved. Okay. Now I will move toward the next problem. Problem number 17. It is related to the Lineweaver-Burke plot, and it says that this Lineweaver-Burke plot is associated with a straight line with a slope of minus Km by Vmax. Number B, a parabola with an intercept of 1 by Vmax. Number C, a straight line with a slope of Km by Vmax. And number D, a parabola with an intercept of minus 1 by Vmax. Just like Michaelis-Menten plot, Lineweaver-Burke plot is very important, one of the very important plots for the enzyme kinetics. Okay. Try to answer this question. I will discuss it in detail in today's class also, and in next week class also. I think we have this enzyme kinetics. There I will discuss this Lineweaver-Burke plot in more detail. So the Lineweaver-Burke plot is associated with what? Is it a straight line with a slope of Km minus Km by Vmax, a parabola with an intercept of 1 by Vmax, a straight line with a slope of Km by Vmax, or a parabola with an intercept of minus 1 by Vmax? Let me check. Okay, there is, yeah, 7/8, it was the correct answer. Good. So yeah, we have two answers. I hadn't checked it earlier, but good, you have done the mathematics correctly. So this was this only a one example of this math. You can be given any of the values there, okay, but just remember to put the values in the equation and you will get the answer. Okay. So here we, for the next question, we have 3 answers and all the 3 answers say it is the number C. Okay, let me check and let us discuss what is the Lineweaver-Burke plot. So the correct answer will be, yeah, it is, sorry, I have just it is the slope of Km by Vmax. It is correct, it is number C. I just misplaced this answer. Okay, I will just correct it here. Yeah, so very good, it is the number C. So what is the Lineweaver-Burke plot actually? What happens? So these are the various equation for the enzyme kinetics. Okay, there is a Michaelis-Menten equation. This is the Michaelis-Menten constant. This is the Lineweaver-Burke equation, and this is the Hans-Wilf equation. So all the equation, sorry, you can reach all the equation from the Michaelis-Menten equation itself. Okay. So, for the Lineweaver-Burke plot, it is a straight line, and in the x axis we have this 1 by S or 1 by substrate concentration, in the y axis we have 1 by V. So, how you get this equation from this equation? How to get this equations? So, what I will do is, I will just reverse it. Okay. From Michaelis-Menten equation, if you solve it from here, we know V equal to V max into S divided by Km by X. If you do it like this, if you take 1 by V, what will happen? 1 by V, it will be then Km plus substrate concentration divided by, we will have what? V max, I am writing it as Vm into substrate concentration. Now, if you just, we can write this equation like this: Km divided by Vm or V max into substrate concentration and plus 1 by V max, where the substrate concentration and it will be cancelled out. We can just divide it like this. Okay. So, if you compare it with a straight line equation, what will happen? We will get y equal to mx plus c, where m is the slope of the straight line, which is your Km by V max. Okay, and the intercept is the 1 by V max. So, this is all about the Lineweaver-Burke plot, and Hans-Wolf plot is similar, just you have to multiply this thing with a substrate of substrate concentration. If you multiply it with substrate concentration, you will get this kind of equation. And the Hans-Wolf plot, it is plotted between the substrate concentration divided by this is the y, this is the y, this is the m, this is the x axis, and this is the c, y equal to mx plus c. So, these are the various equations for the enzyme kinetics. From this equation, from next week we will have some mathematical questions. Okay, I will show you few values of, and you have to calculate what is the particular answer from this using these equations. Okay, so that we will see in the next class for the next week. So, now problem number 18, it says that the fibrous and the globular proteins are associated with dash and dash structure of proteins respectively. Your options are secondary and primary, option B primary and tertiary, option C secondary and tertiary, and option D tertiary and secondary. So, fibrous and globular proteins are two type of protein. So, these are associated with which structures of proteins? Is it secondary primary? Is it primary tertiary? Is it secondary tertiary, or is it tertiary secondary? Try to answer it. Think about the structures of the protein, what is the primary structure, what is secondary, how the tertiary structures are formed, how the quaternary structures are formed. Then think about it. I think you will be able to answer this question. Write in the chat box. So, we do not have much time left for today's class. So, I will just check if somebody has answered it, or I will just, yeah, we have number C. Okay, so let me check if the number C is correct. So, yeah, very good. So, number C is correct. So, fibrous protein is associated with the secondary structure and the globular protein is associated with the tertiary structure of protein. So, see, this is the fibrous protein. Okay. So, when the amino acids are joined in a straight chain to make a fibrous or a sheet like structure, it is called the fibrous protein. Okay. And when the polypeptide chain of amino acids became folded, it makes a globular structure like ring or spherical, then it is called the globular protein. So, what is the particular fibrous and globular protein? So, first one, it is a class of insoluble protein which constitute the main structural element of the body. It is has a long and narrow shape, have a helical or sheet structure in the secondary structure, and it consists of the repetitive amino acid sequence. These are insoluble in water, acid or bases, have a strong intermolecular interactions, less sensitive to changes of temperature and pH, involved in forming the structures like collagen, elastin, actin, myosin, fibrin, keratin, which is present in various fiber of the body, like our hair. Our hair is made of the keratin protein, which is a fibrous protein. Then came to the globular protein. It is a protein soluble in water and tend to involved in metabolic functions, has a round or spherical shape, have a folded ball like structure. In the tertiary structure consists of an irregular amino acid sequence. These are soluble in water, acid and bases, have a weaker intermolecular interactions, sensitive through the changes in the temperature and pH, involved in metabolic reactions, for example enzymes, hormones, hemoglobin, immunoglobulin. Okay. So, this is all about the globular protein. So, now problem number, it will be 19, sorry, there is a typographical error. So, it is the problem number 19. It says that identify the correct statement. Number A, in the interior of globular protein, hydrophilic amino acids are present. Number B, hydrophobic amino acids stay in the interior of the globular protein. Number C, in the exterior, both hydrophobic and hydrophilic amino acids reside. And number D, there is no concrete rule regarding this. So, this was your next problem. It was problem number 19. Identify the correct statement regarding the globular proteins. In the interior of globular protein, hydrophilic amino acids are present. B, hydrophobic amino acids stay in the interior of globular protein. C, in the exterior, both hydrophilic and hydrophobic amino acids reside. And number D, there is no concrete rule regarding this. Take 1 minute if you think you know the correct answer, or if you want to attempt, please write in the chat box. We do not have much time left, so I will just finish it off quickly. So, is there any answer? So, I think there is one answer, number B. Okay, let me discuss it. So, the correct answer is very good, number B, hydrophobic amino acids stay in the interior of globular protein. Now, which amino acids are hydrophilic and which amino acids are hydrophobic? See, if the amino acid is non-polar, means if the amino acid do not have any charge, then it is called the hydrophobic amino acid, like glycine, alanine, valine, cysteine, proline, phenylalanine, tryptophan, methionine, isoleucine, leucine. These 10 amino acids, they do not have any charge. So, these are considered as hydrophobic, and all the charged amino acid like lysine, arginine, histidine, aspartic acid, glutamic acid, these are positively and negatively charged respectively. These are called hydrophilic. Also like serine, threonine, tyrosine, asparagine, glutamine, these amino acids are considered hydrophilic because they are polar in nature. Okay. So, what happens, like in globular proteins are named for their approximately spherical shape, okay, and are most abundant proteins in the nature. The globular proteins exist in enormous variety in 3-dimensional structures. Nearly all globular proteins contain substantial numbers of alpha helices and beta sheets folded in compact nature and stabilized by polar-nonpolar interaction. The globular 3-dimensional structures form spontaneously maintained by result of interaction side chain of amino acid. So, remember this thing. Most often the hydrophobic amino acid side chain are buried closely packed in the interior of the globular protein out of the contact of the water. Okay, and hydrophilic amino acid, they lie on the surface of the globular proteins exposed to the water. Consequently, globular proteins are usually soluble in aqueous solution. Okay. So, remember globular proteins, they are soluble in the aqueous solution. Why? Because hydrophilic or water loving amino acids like this all are present in the surface of the globular protein, and hydrophilic amino acid or water water fearing amino acids, they stays in the interior, making it a compact interior which is out of contact of water. Okay. So, now last question of today's session, it says that in which family of enzyme does RNA polymerase enzyme belong? Number A, ligase. Number B, oxidoreductase. Number C, isomerase, and number D, transferase. In which family of enzymes does the RNA polymerase belong? Ligase, oxidoreductase, isomerase or transferase? So I will just, we don't have much time, so I will discuss it. Okay. So it is number A, ligase. So remember this chart, oxidoreductase, it may be a bit unclear, but when I will share this PPT to you, you will clearly see this. Oxidoreductase is the enzyme which catalyzes the oxidation reduction like lactate dehydrogenase. Transferase is the enzyme which move chemical groups like hemokinase. Okay. Hydrolysis, they catalyze the hydrolysis or bond cleavage with the transfer of the functional group of water like lysozyme. Lyases are the non-hydrolytic bond cleavage like fumarase. Isomerase, the intermolecular group transfer happens like triose phosphate isomerase, and ligases, they synthesize the new covalent bond with using the ATP hydrolysis like RNA polymerase. So remember, RNA polymerase, DNA polymerase, these kind of enzymes are type of ligase. So you don't need to remember each and every name of the enzymes, but try to interpret by their name like what kind of family they can belong to. Like if there is like hydrolysis are present there, the name will actually summarize about it. So few examples I will suggest you to remember for the each and every group of the enzyme for the examples. Okay. And nothing more you will need to remember or memorize. So this is it. It was the last question of today's session. So I will just correct here the number. So today we discussed about the various protein structure, the primary, secondary structure, related question, the enzymes, and all question related to some enzyme kinetics like the globular protein, the what is the fibrous protein, this kind of things we discussed today. And this was your week 3 course content. Okay. So today's session, the questions we discussed today's session, the PPT, the PowerPoint and this video lecture, it will be shared to you by the NPTEL website. So is there any problem or any doubt from the today's session, you can write me in the email. Okay, I am sharing my email ID with you in the chat box. So you can drop an email to me if you have any doubt regarding to today's session. So today was the week 3 session. Like this, every week 3, it was a week 2 session where we discuss the week 3 course content. So like every Tuesday we will have from 6 to 8 p.m. this kind of questions, and I hope today's session will help you for understanding the week 3 course content better, and it will help you to understand what is the week what was the topics for the week 3, and also it will help you to for your week 3 assignment. So was the session okay today? Is there any feedback? I will thank you for joining today's session, and I hope you learned something from today's lecture. And if you have any doubt, any query, please reach me out through the email. Okay, the email ID is mentioned here, and I will request you to join every week's session on particularly regularly on Tuesday from 6 to 8 p.m. Okay. Now if there is no doubt or no query till now, I will just close the session for today, and let's meet on the next week. This lecture video and PPT I will share now with the NPTEL, and they will share it on their web page. Okay. So thank you all for joining. I will close the session for today, and if there is any doubt, please join me, reach me through the email. Okay. Thank you all. Thank you all. Thank you so much. Please join next week's session also.